Hello, welcome to our single-cell multiome analysis series with SciDAP. Previously, we started single-cell clustering analysis, and now we are ready to review the results. In the first tab, you could see the number of cells for each cluster. As you remember, we set 3 resolutions, so now we have results calculated for every resolution we provided. QC metrics for each cluster are shown on the violent plots here. Next, these metrics against each other to see the correlations. There are genes versus RNA reads per cell and RNA reads versus mitochondrial genes percentage. Next tab, UMaps for every resolution and their silhouette score for every cluster. These plots reflect how much the algorithm is sure about to what cluster cells may belong to. The higher the average score, the better. In this case, for example, for resolution 0.3, it is more certain about the cluster 0, cluster 4, and cluster 6 than, for example, cluster 1, or especially cluster 10. If you observe that the average silhouette score is too close to 0, and most clusters have negative scores for the major part of their cell populations, you may want to go back to the settings and rerun the analysis, changing some of the parameters that we've discussed previously. On the tab called Split by Dataset, we see more UMaps, this time for every sample. There are also composition plots that show the same thing, but in a different way. Then there are clusters split by group. We already see that our TRM samples have a cluster that isn't well presented in recirculating cells, as well as the cluster 2 here is not well defined comparing to the other group. With these numbers, you could calculate absolute and relative percentages if needed. Next tab, genes of interest, or marker genes. They split again for each resolution, and their average expression and density are resembled on these two types of plots. These UMaps created for every gene are similar to what you would see in the UCSC cell browser, but already include the axis and the legend. When setting up the run, we provided a file contained 4 example GZ gene sets. This pipeline incorporates tools that calculate gene set expression score per gene list. UMaps, bubble plots, violent plots, all are available for downloading. I only picked 4 gene sets this time, but you could do more. How to prepare metadata for this analysis we discussed in the previous tutorial. Big heat maps with unbiased clustering and normalized gene expression for all genes could be found in this tab. Marker genes that were provided are highlighted here as well. For your convenience, this table containing all the most important information about gene expression in each cluster, including adjusted p-value and log-fold change, could be found here and be exported as Excel or CSV file. Last 2 tabs, the list of downloadable files and the detailed workflow. That was the review of the cluster analysis output. In the next tutorial, we will start working with our ATAC part. Thanks for watching.